It's my distinct pleasure to introduce Dr. Maura Gillison, Professor of Medicine and Cancer Prevention Research Institute of Texas Scholar at the University of Texas MD Anderson Cancer Center. Dr. Gillison is an internationally renowned and accomplished head and neck medical oncologist and molecular epidemiologist who has made seminal contributions to our understanding of the role of viruses in cancer. As a physician scientist, she was the first to definitively delineate the role of human papillomavirus as a cause of a distinct subset of head and neck cancer, resulting in a paradigm shift in concepts of risk, diagnosis, and therapy. Dr. Gillison earned her MD from the Johns Hopkins School of Medicine, then served successively as a postdoctoral research fellow at Johns Hopkins, a medical resident at the Massachusetts General Hospital, the Distinguished Harvard Teaching Hospital, and as a clinical fellow in hematology oncology once again at Johns Hopkins Cancer Center, before earning her PhD in clinical research, clinical investigation at the Bloomberg School of Public Health and Hygiene. As a doctorally trained molecular epidemiologist and physician scientist, her laboratory investigates the implications of human papillomavirus in head and neck cancer, including primary and secondary prevention strategies, diagnostics, prognostics, genomics and molecular therapeutics, clinical decision-making, and population-level cancer incidence in the US and worldwide. As you can imagine, Dr. Gillison is a recipient of numerous awards, most notably including election to the National Academy of Sciences, to the American Society of Clinical Investigation, as well as a recipient of the American Association of Cancer Research Richard and Hinda Rosenthal Memorial Award. Please welcome Dr. Maura Gillison.
Thank you so much. It's a pleasure to be here. I'm a little height challenged, so I have to adjust the microphone. But we've just heard how complex it is to treat an existing malignancy. And I intentionally made the decision to study associations between infections and cancer because there are so many more opportunities to intervene into the disease process. You know, most of the solid tumors we treat have no single necessary cause. The wonderful thing about viral-associated or infection-associated malignancies is there is a necessary cause that provides lots of opportunities to target. So the worldwide burden of virus-associated cancers, it's extraordinarily important research to do in terms of trying to establish infectious disease tumor associations. And we now know that at least 10% of the worldwide tumor burden is caused by viruses in human subjects. The most important of these are human papillomavirus and hepatitis B. And so any conversation about these associations has to start with some of the most important cancer prevention advances in the history of medicine. So that's where I'm going to start the conversation. But the other important aspect of this is the study of viruses and cancers has really promoted a lot of information about how we understand cancers that are not even associated with viruses, given the elucidations of oncogenes through the study of viruses and their role in altering the host cell genome. So I need to emphasize that although we in the United States do have infection-associated cancers, the burden of most of these cancers is in developing worlds. And because of a combination of public health alterations, we have reduced those in the United States, and because of public health advances. The impact of viruses for cancer prevention, I'm going to talk about hepatitis B and HPV vaccines, secondary prevention with HPV detection, and we're starting to see some evidence that cell-free EBV detection may be useful in secondary prevention of nasopharyngeal carcinomas. And then we're going to focus on some of my work, how understanding HPV and its association with head and neck cancers has had profound effects on our understanding population changes in incidence and also in therapeutics for this disease. And then I'll briefly talk about some recent work that we're doing in how we can use the study of the relationship of the HPV virus to the host cell genome to identify drivers in HPV-associated malignancies. So in Taiwan, hepatitis B viral infection is endemic, and it has one of the highest rates historically of hepatoma in children because there's such a high rate of transmission of the virus from the mother to the child in the birth canal. And so one of the most important advances, the first one in terms of public health, was the universal vaccine program in infancy in children in Taiwan, which was instituted in 1984. And very, very shortly thereafter, within 6 years, they started to see dramatic reductions in incidence rates for hepatoma in children. They've recently reported follow-up studies to make sure that that protection lasts later on in adolescence and in young adulthood, and fortunately it does, with 80% to 60% reductions in hepatoma rates in vaccinated individuals. So most of the failures that they see are in children who either are not vaccinated or who don't receive all 3 vaccines or whose mothers are very heavily infected and transmit to children. So this, I think, is a tremendous public health advance in understanding and how it can impact population-level disease. With regard to another virus type, HPV, HPV was identified, or papillomaviruses were identified as the first infectious agents that could transmit from one subject to another and alter the growth of a human cell in the early 1900s, subsequently identified as HPV. And it really is to the benefit of people, largely gynecologists and their colleagues who are mostly epidemiologists, how understanding the natural history of HPV infection provided lots of opportunities for intervention. So when Harold Zerhausen identified HPV-16 in a cervical cancer in the late 1970s, because of the recognition that HPV had induced cellular changes early in the 1930s and instituting the Papa-Nicolau smear in the 1950s, even before HPV was recognized as a cause, it allowed epidemiologists to piggyback studies onto an existing infrastructure in clinical medicine, the Pap smear. And it became clear that HPV infection was necessary for the development of cervical preneoplasia. And it's really the individual who does not clear their HPV infection within a couple of years of initial infection that remains at risk for the development of the cancer. And this provides an opportunity for detection of the HPV and treatment. And then, so it's really these persistent individuals who are infected. And as a result of the identification of HPV, Doug Lowy and John Schiller were able to demonstrate in vitro that the HPV L1 viral particle, the capsid protein, has the ability to self-assemble, which led to the development of the HPV vaccines. I think the second most important cancer prevention advance in the history of medicine was the introduction of the HPV vaccines. And just recently, in the last month, the final study, the final report of the HPV-9 versus HPV-4 vaccine was published in Lancet. The HPV-4 vaccine covers HPV-16 and 18, which account for about 70% of cervical cancers worldwide. The benefit of the HPV-9 is it increases that to about coverage of 90%. And in these trials, there were 14,000 women that were randomized to receive the HPV-9 versus HPV-4 vaccine. And then they were followed up subsequently, and this is the report of 6-year outcomes in that trial, which showed about a 98% efficacy in the reduction of both incident infections by viral types, and then, of course, high-grade cervical disease. So very important, and we're starting to see in regions where governments made the decision to provide the vaccine for free in schools. For instance, in Australia, very high herd immunity and very high uptake. And they're starting to see at the population level first reductions in HPV infections, and then reductions in genital warts caused by 6 and 11, and subsequently reductions in CIN3, high-grade cervical lesions, in those populations, which are extraordinary. The identification of HPV and HPV persistence as indicators of individuals who are at risk rapidly resulted in clinical trials comparing HPV detection and its effectiveness in screening in comparison to the historical cervical cytology. And these graphs show the results of summary kind of a meta-analysis of 4 different trials involving 170,000 women, demonstrating for the first time that a single detection of a positive high-risk HPV test subsequently reduces the incidence rates of cervical cancer in individuals who have evidence of prior exposure by 60%. And then in individuals who have an HPV negative test at baseline, showing a 70% reduction in prospective diagnosis of cervical cancer. So remarkable combinations now of cervical cancer screening and HPV vaccines can reduce cervical cancer incidence dramatically in populations. Now in my work, this slide summarizes the first decade of my career. What we were able to do is, in collaboration with a number of my colleagues at Hopkins, is demonstrate that HPV was present in about 25% of head and neck cancers at this time, and that it seemed to have a distinct clinical presentation of tumors that arise largely in the tonsil, the base of tongue, and in the palate. And that these tend to occur in younger individuals without the traditional smoking and drinking risk factors that I was taught about in medical school as the cause of head and neck cancer. They tended to have a specific histology, and then we went on to show that they have unique risk factors. But why does this matter? At the time when I made this initial observation, I thought, well, this could be a very interesting observation, but perhaps irrelevant if it doesn't have an impact in terms of what's happening in populations. And so this allowing us to distinguish a cause of a cancer that was from a virus versus from alcohol and tobacco subsequently allowed us to understand its implications for populations. Now, rates of smoking in the US had started to decline in the 60s because of the Surgeon General's warning of relationship between tobacco and lung cancer. And yet it was noticed in the 90s that head and neck cancer incidence rates were still going up in the United States, which didn't make a whole heck of a lot of sense. And so once we had established HPV as a cause of a subset of head and neck cancer, we did this work in collaboration with the National Cancer Institute looking at, well, what impact is this having at the population level? And indeed, oropharynx cancer incidence rates were going up. So we obtained all of the head and neck tumors that had been collected over 2 or 3 decades by the National Cancer Institute and tested them in my laboratory for HPV. And what that allowed us to identify is in a only 16-year period of time, as shown here, the rates for HPV negative head and neck cancer, largely driven by smoking and drinking, had declined indeed in that 16-year period by over half. But unfortunately, at the same time, the HPV positive disease had increased by 225%. So that the cancer we were treating largely in the 1990s was the smoking drinking cancer, but just 16 later, the predominant form of this cancer we were treating was actually HPV positive head and neck cancer. Why did this matter in terms of incidence and therapy? Well, we were starting to see the tip of the iceberg in terms of an epidemic that is being driven by oral HPV infection and largely in men. And we've gone back to look at prospectively the SEER data, and this is showing the incidence rates for oropharynx cancer by age in men versus women. So you see very few cancers where women and men share the same anatomy. Do you see these incredible differences? And we've gone on to show, and people are demonstrating this prospectively, that this is because men don't seroconvert and develop protective antibodies in response to genital HPV-16 infection, whereas women do. So we've been targeting vaccine strategies to women when men are actually kind of the typhoid Marys, per se, of HPV. They carry the virus, and they don't develop their own protection afterwards, so they remain at risk, which is why we're seeing these dramatic increases in head and neck cancers caused by HPV in men and largely not in women. As a result of the sexual revolution, women became genitally exposed, developed high titers of serum antibodies that subsequently protected them against cervical oral HPV infection, but men do not. The other impact of understanding that there was a subset of head and neck cancers caused by HPV is we demonstrated in the context of clinical trials that the HPV status of your tumor is the single greatest predictor of whether you live or die after a diagnosis of head and neck cancer, with HPV-positive patients doing much better than HPV-negative patients. So we developed all the assays that are clinically current standard of care in classifying patients as HPV-positive or negative, and the clinical trial approaches to these cancers have markedly diverged. We also showed at the population level that this was also true, that in SEER cancer registries, HPV-positive patients have a remarkably improved survival over HPV-negative patients. And we'd also shown in clinical trials that smoking modifies the survival outcome of patients with HPV-positive disease, so that the more you smoke, even though your tumor is driven by HPV primarily, the less well you do with current standard of care therapies. But we had seen at the population level, and we thought this was because of our improvements in therapy, that their survival for head and neck cancer was improving over time. Well, recall we saw a dramatic shift in the incidence trends for HPV positive and negative cancers over that period of time when we were intensifying head and neck cancer therapy in the clinic. And we thought at the population level that this was having an impact on survival in head and neck cancer. Well, once we had the ability to find and identify HPV positive versus HPV negative cancers, we were able to go back and look at this data again and show that unfortunately we'd made no inroads in the therapy of HPV negative head and neck cancer. What was actually happening is HPV positive patients were smoking less. We didn't recognize them as an HPV positive disease because they were smokers. Smoking was so common. But as they decreased their rates of smoking, HPV positive patients were presenting with head and neck cancer who'd never smoked, and their response to chemotherapy and radiotherapy was markedly better. So it was that subset of the population going up over time and their reductions in tobacco use that was actually driving the improvements at the population level and survival, and it wasn't the therapy, unfortunately. So it gives you an example of how understanding the relationship between an infection and the cancer not only provides you opportunities to understand and target and prevent a particular disease, but it allows you to understand population level shifts in cancer that can be quite extraordinarily important and can happen in very short periods of calendar time, only 20 years. So now I'll talk to you about a little bit more about my recent work, because in cervical cancer we know a lot about the natural history of and risk factors for acquiring HPV, about the natural history of HPV infection, and risk for progressing to some extent. But what has been a real black box is, although HPV and the functions of E6 and E7 are clearly necessary in the development of cervical cancer, the secondary genetic events that actually promote the development of invasive cervical cancer have really been a mystery. And there have been recent publications of the cervical cancer landscape looking at, through whole exome sequencing, mutations that occur that are greater than by chance in these tumors. And we've done the similar work now for HPV positive head and neck cancer in my laboratory. And this is the result of that work, the genome landscape of HPV-positive head and neck cancer. And it is indeed, like the epidemiology of the cancers and the incidence trends in response to therapy, a unique disease from a genetic standpoint. And this is the best way I can show that. What you see on the top is mutations that are significantly enriched in HPV-positive cancers, represented in red, in comparison to those that are most frequently altered in HPV-negative cancers in blue. And on the bottom you see vice versa. So you see there are only a couple of genes that are shared in terms of frequent mutations in the two cancers. So clearly genetically distinct. But what we did while we were doing this is some work previously in cell lines, which frankly I did as someone who's a molecular epidemiologist. I needed to make sure that I could do genomics. I became a little bit more comfortable with the idea that it's just descriptive statistics in very large data sets, which I was kind of used to doing. But I wanted to make sure I could do it. And so we acquired all of the HPV-positive cell lines that existed and did whole genome sequencing on them, and discovered something kind of incidentally while we were doing that related to HPV integration. So HPV is actually a very small double-stranded DNA circular virus. It's only about 8,000 base pairs. And normally upon infection, it exists as an episome in the nucleus of the infected cell. And it was known based on some assays that were kind of archaic, but nevertheless, that with the histopathologic progression of cervical cancer, you saw an increase in the rates of integration into the host cell genome. And it had been shown that that results in chimeric transcripts that stabilize the transcript and increase E6 and E7 expression. And it was kind of the dogma that the E2 negative regulator of the E6 and E7 promoter also is frequently disrupted during integration and results in dysregulated E6 and E7 transcripts. So basically that the role of integration was just to stabilize the E6 and E7 transcript. And so when we did this first cell line experiments, we developed the ability to identify the viral host cell genome chimeric sequencing reads. And then we mapped those to the host cell genome, and we found what we called clustering of these viral integrants. At first, when we first saw this Circos plot, we said, oh my gosh, we're identifying hotspots. But then when we looked more carefully, it turned out that each of the individual cancers, these reads were in individual cancers, and they weren't across multiple cancers. But what you can also see in these Circos plots, which we were quite struck by, is these sites of integration directly flanked regions of the genome with copy number variation. So we were very intrigued by that, and in a collaboration with investigators at NCI, we actually used FISH and SCI and showed that these regions also frequently flanked chromosomal translocations. So when we looked directly at the IGVs, what you're seeing here is CHA cells and HMS001. One's a cervical cancer cell line, one's a head and neck cancer cell line. And we've taken the whole genome sequencing reads in the control, which is yellow, and in our tumor sample, in this case cell line, which is blue, and we superimpose them so a region that is amplified will be blue, a region that is deleted in the sample will be yellow. And so what you can see here, and then the sequence, the chimeric sequence reads that are virus and host cell genome, which are the integration breakpoints down to the base pair, are indicated in red here. And what we saw repeatedly is that the viral host chimeric sequencing reads mapped directly to the amino acid, to the base pair, to regions of copy number variation and structural variation. And these are simple examples here, but they can actually get quite complex, where we see the virus in the top cell line directly flanking a region that has, I think, about 180-fold amplification of a specific region of the genome. Below that, you see a region where the viral sequences directly flank a large region that is deleted. And then these can be quite complex and quite large regions of the human genome. And there seem to be steps within steps of these. We're not quite sure if this is clonal evolution or looking at what the integration events happen cumulatively over time in the same cell. We're now trying to understand that with single cell sequencing. But what we ended up coming up with once we identified these abnormalities, we actually went on this chromosomal walking expedition, and some of these things took 1,000 primer pairs to resolve, but we started with this easy one, which is in CIHA. And what you see here is the host genome. We've taken host rearrangements and indicated those, and we've also taken the viral breakpoints and indicated those. And then we fragmented the reference genomes into these segments defined by those. So you see on the top, in the reference genome, A, B, C, D, E are the fragments. And then the virus is always represented in red. And when we sequenced across the whole region, what we observed was the resulting structure actually in the cell was A, B, C, D, and then 7,000 base pairs of viral sequence going back to B. Then there was a host-host rearrangement, B to D, with the C region deleted, and then D, E, and out. So while we were drawing this, what you end up having is these host virus genome concatenamers that are in the genome. And some of these regions we see 180, 200 of these host viral concatenamers right in a row. And so what we had hypothesized, since the virus HPV doesn't encode its own machinery to replicate itself, it actually uses ATM and ATR triggering to replicate itself. So in response to DNA damage, we think the virus has ATM and ATR recruited to its origin of replication, and you can actually get aberrant rolling circle viral amplification that results, the virus actually picks up part of the host cell genome and results in this amplification, and then during repair it gets back inserted into the host cell genome. So not only do you get basically amplification of the viral sequences in E6 and E7, but you also get amplification of the intervening host genomic DNA. And what we found is the regions that are deleted frequently had tumor suppressors, and the regions that were amplified frequently had oncogenes. So we're no longer doing this painful chromosomal walking. We're now experimenting with the help of people at the New York Genome Institute, long-read sequencing methods to try to resolve these structures. And this is the example, and we're also looking at implications of the viral integration events on host genome transcription. So this is an example in which we identified HPV amplifying by several-fold the FOXE3 gene, and we confirmed that with long-read sequencing, our hypothesized structure, which is in B, based on our looping model, and then verified that that was indeed the case in the long-read seq. And then through RNA-seq, we show that there's dramatic upregulation of FOXE3 in this tumor. And this is in a primary tumor now, no longer cell lines. So we've now done about 100 of these tumors, and we're analyzing what are we actually seeing in the tumors themselves. What we've shown in cell lines is that we see amplifications and deletions. We see those in humans as well, in the primary tumors. This isn't an aberration of cell culture. We see rearrangements. We see HPV mediating translocations. We see HPV insertion of super enhancers, and we verified that in the laboratory. We see gene breakage, and we even see some cases in our cell lines where the HPV generates antisense transcripts, which subsequently affects protein translation. So as we sequence more and more tumors, what we're finding is repeated events of particular genes, and this is an example of that. Recurrent HPV looping at the MYC locus. We see amplification of a region of the genome in an intergenic region, either upstream or downstream of MYC, but in some cases we actually see, as here in the bottom line, actually amplification of the MYC gene itself and associated increased expression of MYC by RNA-seq. We've actually used CRISPR-Cas9 now to knock in just concatemers of the HPV promoter within 500 kb in either direction of MYC, and shown that that can mediate marked increase of MYC expression. So we think this is basically the viral promoter acting as an enhancer and resulting in increased expression of MYC. With collaborators in the Schlegel lab, we've had cells grown out from tumors and shown through lentiviral shRNA knockdown. The belief in the field is E6 and E7 expression is always necessary for the malignant phenotype. At least in this one patient now, we've shown that the driver has switched from E6 and E7. If we knock down E6 and E7 in the cell line, the cells still grow, but if we knock down MYC, they don't. So our hypothesis now is that and we've shown that these integration events are indeed enriched in open chromatin, fragile sites, et cetera. But our hypothesis is that this could drive clonal selection. And through the study of viral integration, we may be able to identify drivers in these tumors. So that's why we're doing it in large scale now. But we've also developed a model system whereby we actually transfect human tonsillar epithelial cells with the virus as a kind of plasmid, and then culture it over time. And this is our first example where we found a dominant clone in the culture, and we took it out and sequenced it, and it turned out that the virus had integrated near MYC. And actually resulted in 16-fold amplification of MYC, clearly driving the clonal selection of this particular clone in the cell culture. So I think I've shown you why it's so important to understand and study viral infectious tumor associations. Not only does it allow you to identify known for very few cancers, a single necessary cause that allows you to intervene through improvements in public health, development of vaccines like hepatitis B and hepatitis and HPV, seeing at the population level the impacts of those vaccines. It allows you to screen for people at risk and reduce the subsequent incidence rates for those cancers. I think we're going to start to see that also for EBV. Most people don't know that the EBV field, the nasopharyngeal carcinoma field, has been leading cell-free DNA as a biomarker of risk of progression for about 20 years now. And now, published recently in the New England Journal, we may be able to use cell-free DNA and EBV persistence as a measure of in endemic populations for identifying people who have this malignancy. And then I've shown you, we've known for 100 years that the study of cancers that are virus-driven has really informed a lot about non-virus-associated tumors in terms of identification of oncogenes. And now in the era of genomics, I've shown you how the study of the relationship between the viral genome and the host may allow us to inform the secondary genetic events that are important for these cancers to develop. And I can say, in collaboration with individuals at Harvard, we've now shown that Merkel cell virus does the same thing as does hepatitis B virus. So it seems to be something that might be common. So I thank you so much for your attention. It's been an honor to talk to you today. Thank you.
Thank you so much for that really magnificent talk. We have time for a couple of questions. Yes, please.
I was wondering, there are reports on association of breast cancer with papillomavirus, and if anybody has done, has looked on the incidence of breast cancer in vaccinated with papilloma vaccine and non-vaccinated women, whether incidence of breast cancer are lower.
Right.
Yes.
Well, at some point or another, HPV has been hypothesized to cause almost every human malignancy. And I think in large part, the studies that haven't ended up being necessarily true are that it's really easy to do HPV detection assays, but somewhat difficult to do them well in order to prevent, laboratory contamination of the specimen. And so the question about HPV's relationship to breast cancer has come up quite a lot, as has Epstein-Barr virus. And I think those associations in prospective studies, like you're saying, haven't really fallen out. But you're right in terms of and that's why the CDC is doing such important prospective surveillance trials of cancer incidence before and after the introduction of the vaccines, as are many countries, because that's, in Koch's hypotheses, that's the ultimate way of showing an association is if you, intervene with a vaccine, seeing subsequent reductions at the population level. So I think you're right. Such surveillance studies are really important, and they may inform, for instance, in hepatitis B, the chronic inflammatory state caused by chronic hepatitis B infection has been hypothesized to increase rates for other cancers. Perhaps in populations that get the hepatitis B vaccine, we'd be able to see that at the population level.
Andrew?
Yeah, so this is somewhat related questions. We had a lot of discussion in the last panel about the emergence of resistance in cancers treated with therapies. I'm wondering just your feeling for the HPV vaccine, specifically the 9-valent one. Do you think that's actually just pretty much nailed it and it would be almost impossible to evolve resistance, or for viruses to evolve that would be resistant to that? Or do you think that escape from that vaccine would be possible? Sort of some virus that could actually still be an effective infectious HPV, but not be inhibited by that vaccine.
So I think in that, that would require. Well, first of all, HPV is a pretty stable virus.
Yeah.
So
DNA.
it's a DNA virus, not an RNA virus. So it doesn't have the ability to mutate itself quite as readily as an RNA virus. The other thing is that the capsid structure and its alteration with binding has been critical to show in uptake of the virus. So you'd have to hypothesize that there could be a mutation in the capsid that could promote evasion of those antibodies and yet at the same time still have conformational changes that allow uptake. So I think, theoretically that's possible, but I think it's probably unlikely. Certainly that's another reason why to do surveillance, but so I think that's a theoretical concern.
But practically, it sounds like, from a public health standpoint, if you could effectively vaccinate populations, at least to get even the least effective herd immunity, then you really have solved the problem for all these cancers that are caused with
yeah. I think the hurdle is having governments and cultures make the right decisions about instituting the virus and the vaccination in the first place. But there are extraordinarily effective vaccines. Yeah. It's been hard to study resistance to the vaccines because we can't find people who fail them. So, fortunately.
Okay, thank you.
So I'd really like to thank Dr. Gillison for really a superb conversation and for the audience's participation.